chromosomes attached to those ropey microtubules right in the middle at their centromeres. The chromosomes then begin to be moved around, and this is being done by molecules called motor proteins. And while we don't know too much about how these motors work, we do know, for instance, that there are two of them on each side of the centromere. These are called centromere-associated protein E. So these motor proteins attached to the microtubule ropes basically serve to spool up the tubule slack. At the same time, another protein called dynein is pulling up the slack from other ends of the ropes near the cell membranes. After being pulled in this direction and that, the chromosomes line up right down the middle of the cell. And that brings us to the latest installment of biologically. So how do chromosomes line up like that? We know that there are motor proteins involved, but how? What are they doing? Well, remember when I said earlier that there were a lot of things that we don't totally understand about mitosis? It's sort of weird that we don't, because we can literally watch mitosis happening under microscopes. So chromosome lining is a good example of a small detail that has very recently been figured out. It was a revelation 130 years in the making. Mitosis was first observed by a German biologist by the name of Walter Fleming, who in 1878 was studying the tissue of salamander gills and fins when he saw cells' nuclei split in two and migrate away from each other to form two new cells. He called this process mitosis, after the Greek word for thread because of the messy jumble of chromatin, a term he also coined that he saw in the nuclei. But Fleming didn't pick up on the implications of this discovery for genetics, which was still a young discipline. And over the next century, generations of scientists started piecing together the mitosis puzzle by determining the role of microtubules, say, or identifying motor proteins. Now, the most recent contribution to this research was made by a postdoctoral student named Tomomi Kiyomitsu at MIT. He watched the same process that Fleming watched and figured out how at least one of the motor proteins helps snap the chromosomes into line. He was studying a motor protein called dynein, which sits on the inside of the membrane. Think of the microtubules as tug-of-war ropes, with the chromosomes as the flag in the middle. What Kiyomitsu discovered was that the dynein plays tug-of-war with itself. Dynein grabs onto one end of the microtubules and pulls the tubules and chromosomes toward one end of the cell. When the ends of the microtubules come too close to the cell membrane, they release a chemical signal that punts the dynein to the other side of the cell. There, it grabs onto the other end of the microtubules and starts pulling until smack, it gets punted back again. All of this ensures that the chromosomes will line up exactly in the middle so that they will be split evenly. That discovery was published in February of 2012, a couple of weeks before I sat down in this chair, and 134 years after mitosis was first observed. If you want to join the ranks of scientists who are answering the many questions left about mitosis and lots of other things about our lives, maybe someday I'll do a biography about you. Now, so far we've gone through the interphase, where the centrosomes in DNA replicate themselves and get ready for the split. The prophase, where the chromosomes form and the centrosomes start to spread apart, in the metaphase, where the chromosomes align in the middle of the cell. And now, it's time to separate the chromosomes from their copies. This time, motor proteins start pulling so hard on the ropes that the X-shaped chromosomes split back into their individual single chromosomes. Once they're detached from each other, they're dragged toward either end of the cell. Since the prefix "ana" means back, that may help you remember the name of this phase, called anaphase. After this, it's just a matter of using all that genetic material to rebuild so that the copied genetic material has all the accoutrements of home. In the last phase, telophase, each of the new cell structures are reconstructed. First, the nuclear membrane reforms, the nucleoli form within them, and the chromosomes relax back into chromatin. And a little crease forms between the two new cells, which marks the beginning of the final split. That division between the two new cells is called cleavage, and all that's left is to make a clean break. This is done by cytokinesis, literally cell movement, by which the two new nuclei move apart from each other and the cells separate. We now have two new cells, each with a full set of 46 chromosomes. These clones are called the daughter cells of the original cell.